Welcome to our video lecture on genes. Our essential understanding for today is that every living organism inherits a blueprint for life from its parents. Our objectives: we're going to define gene and genome. We're going to talk about genes and alleles. We're going to explain the cause and the consequences of sickle cell trait. We're going to explain that alleles are formed by mutation. We're going to describe the Human Genome Project. We're going to comment on some of the ethical issues related to bioinformatics, and we're going to wrap up with a quick look at DNA sequencing. So just a really beautiful graphic showing us this idea of human genome being the blueprint of life. So we've got chromosomes in our nucleus. Those chromosomes are composed of DNA. Genes are pieces of DNA that code for a trait. Those traits influence who we are. So gene defined. Gene is a heritable factor that consists of a length of DNA and influences a specific characteristic. This is the gene sequence for human insulin. Just the A's, C's, T's, and G's that make insulin. It's kind of cool that we know that, right? Genes occupy specific positions on one type of a chromosome. That position, that location, is called the locus. Loci is plural. Those locations are the same on homologous chromosomes. What's kind of cool is that so we get chromosomes in pairs from our parents. We call those pairs homologous chromosomes. The homologous chromosomes will have the same genes at the same loci. So we get hair color genes from each of our parents. So hair color might be here and here. Not necessarily the same version of hair color, but that gene, one from mom and one from dad, will be in the same place on these homologous chromosomes. Here's a map of chromosome number 7 for us humans. Centromere is here in the center-ish. The short side off the centromere is the P arm, and the long side off the centromere is the Q arm. And this is just showing a few of the genes on human chromosome number 7. We have beta chain antigen receptors. This is part of our immune system. Interferons are also part of our immune system. Epidermal growth factor related to growing skin. Hormone for red blood production. Here, interestingly, is the mutation that causes cystic fibrosis. The genome is all the genetic information of an organism. So for us humans, it's 46 chromosomes in our nucleus, but also those chromosomes in our mitochondria. So mitochondrial DNA is also part of our human genome. We mentioned on the previous slide that you get the same genes from each of your parents, same genes on the same chromosomes and the same locations, those same loci. But we don't necessarily get the same versions of those genes. And those different versions of genes are called alleles. So alleles are different forms of a gene. They are going to be in the same locus on the same chromosome. Most of us, plants and animals, have 2 copies of each gene. So 2 alleles, one from mom and one from dad. What's pretty cool is that those alleles, even though one allele might code for blonde hair and one for black hair, a different one for brown hair, those alleles of the same gene differ by only a few base pairs, so only a few A's, T's, C's, and G's different. We call those differences SNPs, or single nucleotide polymorphisms. Single, just one. Nucleotide, those A's, T's, C's, and G's. Polymorphism, poly means many, and morph means shape. So we have many different shapes of these differences, these SNPs. Here's one example of a SNP. So we can see these different alleles, one dominant, one recessive trait, different alleles for the same gene. And notice that they're at the same gene locus. We got one of these chromosomes from mom and one from dad. We don't necessarily know which one is which, but one of those came from mom and one came from dad. Same genes, but not necessarily the same alleles in the same locations. So these alleles are formed by mutation. Mutations are changes in the DNA sequence. We're just flipping around some of those A's, T's, C's, and G's. Mutations are random, though there certainly are factors that can influence the rate of mutation, but what those changes in the DNA sequence are is quite random. It doesn't happen because we need them to happen. Mutations just happen. Some of them are good and some of them are bad. Base substitution is one kind of mutation, and this is actually the mutation that results in sickle cell trait, which is what we're looking at in this graphic here. IB really find sickle cell trait interesting, and so make sure that you know this information about sickle cell trait. So wild type, or the normal DNA code for hemoglobin. Hemoglobin is the protein that carries oxygen on our red blood cells. This is the normal DNA sequence, just at this little piece of the hemoglobin gene. So we have CTC, cytosine, thymine, cytosine. It's going to code for messenger RNA, guanine, adenosine, guanine. This is the codon in the messenger RNA, the normal codon, and that's going to lead to this glutamic acid in the normal version of hemoglobin. So this is the amino acid in this particular spot of hemoglobin. If we have just one tiny little change in the DNA sequence of the gene for hemoglobin, if we flip from thymine to adenosine, then we're going to have a different codon in the chain of messenger RNA. We're going to flip that adenine for a uracil, and that's going to code for a different amino acid at this one particular location in hemoglobin. So we have valine instead of glutamic acid. This one different amino acid in this sequence of amino acids in this polypeptide chain causes shape changes on the red blood cells when oxygen levels are low. That shape change looks like this. Red blood cells ought to be these donut shapes that travel through our blood vessels quite easily. When a person with sickle cell is in an environment of low oxygen, the red blood cells will sickle or turn into these crescent shapes. And what happens is they catch on each other. And so we get these clots of red blood cells, which of course is going to prevent the carrying of oxygen and nutrients to the rest of the body that's downstream of this clot. Lots of tissue damage and lots of pain for these people with sickle cell. It is a terrible disorder. So we know the code for sickle cell trait. We know lots of codes. We know so many codes. We actually have a whole field now related to this data that we have regarding DNA and messenger RNA and protein sequences. And we call this field bioinformatics. It's kind of like the IT department, the IT field of biology. One of the first sources of information for bioinformatics was the Human Genome Project. So this was started in 1991. It was an international collaboration, 20 different universities and research centers in lots of different countries working on sequencing the whole entire human genome, so all of our DNA and all of our chromosomes, trying to figure out the A's, T's, C's, and G's that made humans human. So interestingly, it happened faster than expected. We finished sooner than planned, and also under budget, which was super cool and, again, unexpected. And part of it was because of the broad array of perspectives in different cultures that we had because of this international collaboration, but it was also because a lot of competition between these guys in particular. So Craig Venter, Francis Collins, Collins directed the Human Genome Project for a while, and Venter, was like, I can do it better. And their competition actually led to some pretty amazing advances in technology that allowed the human genome to be sequenced more quickly and less expensively than was expected. Another crazy thing that happened, there were fewer genes in the human genome than expected. We had a lot of DNA that didn't code for proteins. And at that time, we called it junk DNA. When I was in college, I learned junk DNA. But now we actually know that a lot of this quote-unquote junk does really cool stuff that isn't necessarily coding for a protein, but it does other important things. And then one more cool guy. So this is Skip Garner. Skip Garner was co-director of the Human Genome Project, the piece of it that was happening at UT Southwestern in Dallas. I was very lucky. I got to work with Dr. Garner in his lab at UT Southwestern as a summer intern, so it was pretty exciting. Here's just a tiny little graph showing a little bit of one of those amazing consequences of the Human Genome Project. The cost to sequencing human genomes dropped dramatically. So here's 2001. It was about $100 million to sequence human genomes. Now it's down into the hundreds. So this graph ends at 2019. We're sitting right at about $1,000, which is way different from $100 million. And a lot of it is because of that Human Genome Project, those advances that were made that just allowed us to do things faster and cheaper. So other little pieces of bioinformatics, just some cool stuff. There was a company a few years ago that decided they were going to sequence people's genomes for free. So you would submit your DNA sample, they would sequence it for free and give you that information. The caveat was that you had to allow your DNA to be put into a database and be used for research. So noodle on if you would be okay with that, if you'd be okay with your DNA being accessible to researchers and insurance companies and who knows who else. Another piece of bioinformatics, now you can spend a couple $100, get your DNA sequence, and it'll tell you how to be healthy given your DNA. It'll tell you some of the things that you should eat, some of the things that you shouldn't eat, how you can be healthy with your particular DNA. So that's kind of cool. Another cool thing, so Ancestry.com, of course, has been around for a while. This guy right here, this is my dad. My dad did Ancestry.com quite a few years ago. My dad was adopted as a baby. He did not know his birth parents. He's been looking for them for a while. A couple of years ago, he gets a letter in the mail from Ancestry.com. They found his dad, and this is his dad. This is my Gramps. So I've known my Gramps now for 3 whole years. And I just gotta say, genetics is pretty cool. These guys look a whole lot alike, right? The craziest part to me, and I said this in class, but I'm gonna say it again, they dress alike. Like, this is the weirdest thing. Like, how is this genetics? So this is my dad, and every single day, he wears this short-sleeved, button-down, usually a pale blue, green, yellow colored shirt. He's always got something in his pocket. He wears a brown belt, jeans, and brown loafers. I would like you to look at what my Gramps is wearing. My Gramps is wearing a short sleeve, button down shirt in a palish color. He's got stuff in his pocket. You know what else he's wearing that you can't see in the picture? Brown belt, jeans, and brown loafers. How is that genetic? Crazy sauce. They have the same outfit. Amazing. So, less amazing. So, quite a few years ago now, this genetics company decided they were going to patent Angelina Jolie's genes. So, Angelina Jolie, actress, she tested positive for a couple of the genes that lead to higher risk of breast and ovarian cancer. And so, this company decided that they were going to patent her genes. Because, you know, why not? And then the tests for those cancers, they would own the sequences that allow people to test for those cancers. They owned the patent for Angelina Jolie's DNA sequences. Luckily, the Supreme Court came in and said, no, you cannot patent human DNA sequences if they naturally occur. If you make up your own sequence, sure, but human DNA sequences that just happen in humans can't be patented. So there's that at least. Another little, less wonderful story regarding bioinformatics. So a lot of our research done in genetics is done with these amazing cells that are called HeLa cells. So HeLa cells come from Henrietta Lacks. This is Henrietta Lacks. So Henrietta Lacks, HeLa cells. So these HeLa cells are cancer cells. Henrietta Lacks died of cervical cancer, and doctors harvested some of her cervical cancer cells without her knowledge or permission and started growing them in their lab, and it was amazing. They were astounded because these cells kept growing. They were, they wouldn't die. And so we have over the last 80-ish years, we've grown about 50 million metric tons of these cells. 50 million metric tons of this woman's cervical cancer cells without her permission. All of these patents, so many research articles, at least 4 Nobel Prizes directly related to research on her cells, and a bunch more that are indirectly related. Her cells have been to all 7 continents and to outer space. Her DNA has been sequenced and published for the whole entire world to access. She did have family, and so her family's DNA is therefore also out there for all the world to see. We have made developments in treating and preventing polio and HIV, Ebola, sickle cell, HPV, tuberculosis, salmonella, parvovirus, which is the pet version of HIV, cancer, and most recently, COVID. So COVID vaccines have also been tested, developed using her cells. It's sold for about $250 a vial. And guess how much money her family gets from this? That would be $0. $0, my friends. And this was all done without her permission. But she was a poor woman. She was an African American woman. So the doctors maybe didn't feel like it was all that necessary. It's a super interesting story. So I highly recommend that you look into some research into Henrietta Lacks. Noodle on the ethics of this. So in the last couple slides, we've talked a lot about this information regarding DNA sequences that we have, all this information in databases. So let's talk about how do we even get that information? How does DNA sequencing even work? So what's going to happen is we're going to take our strand of DNA. Maybe this is a strand that we want to code CTGA. I don't know what the code is yet, but I want to figure it out, right? I'm going to take this DNA. I'm going to have lots of copies of it. We do a super cool thing called PCR, polymerase chain reaction, to make lots of copies of it. In our PCR, what we're going to do is have lots of A's, T's, C's, and G's so that we can make those copies. We're going to have some polymerases that will allow us to bind together those A's, C's, T's, and G's that would be complementary to our DNA sequence. But we're going to add in each of these different flasks is one extra special dideoxynucleotide. Remember that RNA is ribonucleic acid, right? Because it has the sugar ribose. DNA has deoxyribose for the sugar. So now what we're going to add is dideoxy. So instead of just taking off 1 oxygen, we're going to take off 2 dideoxy. So we're going to take off 2 oxygens. And what's crazy about dideoxynucleotides is that we cannot bind anything to them. So polymerase can like work along. If it gets to a dideoxynucleotide, in this case this G, replication is done. It can't go any further. So what we're going to do is we're going to take this DNA sequence and I'm going to add it to each of these 4 different flasks. All of those flasks have normal A's, T's, C's, and G's. All of them have normal polymerases and all the other stuff that we need for DNA replication. This flask only is going to also have dideoxythymine, and it's going to be radioactively labeled dideoxythymine. Same thing here, except now it's going to be dideoxyadenine, and here guanine, and here cytosine. What's super cool about that dideoxy stuff is that, again, it does stop that replication. So in this flask, every once in a while, we're going to get to a T in that complementary strand, and replication will stop. And in this flask, every once in a while, we're going to get to an A in that complementary strand, and replication will stop. And then what I can do is I can put it into our gel electrophoresis, and we're going to separate things by fragment size. So this fragment that stopped at the T, because it happened to be that dideoxy T at this location, is going to be super small and it's going to travel to this distance in the gel. So I'll know that I have a T in the sequence. The next shortest strand will be a C. We'll know that we have a C in the sequence. And then we have a G, and then an A, and then an A. A G, an A, an A. And then a G, another G, and then a T, and then a C, and then an A, and then a G, and then a T, and then a C, and then an A, and then a G. So we can read from which wells we put these dideoxynucleotides into. We can read the fragments, the last nucleotide in the fragments, and we can figure out what the DNA sequence is, which is super cool. Now we can actually just use a sequencing machine and label those A's, T's, C's, and G's, and the computers will just read off different signals from the bases. And so instead of a human having to go, oh, first it's T and then it's G and then it's G, or sorry, C and then G and then an A, instead of a human reading that and perhaps making a few errors, the computer can just do it crazy, crazy fast, which is better. And here's just a quick look at that dideoxy structure. So here we have ribose. Notice I've got OH, deoxyribose. I have an H here because I took off that O. Here I have dideoxyribose. I took off 2 oxygens. There's no oxygen in either of those spots. And then just one more look here. Again, we're going to read T and then C, and then G, and then A. And this is possible because of those dideoxy nucleotides that we added to these different flasks. If you are interested in bioinformatics, if you're like, ooh, I love this big data, there are free online public accessible websites, databases that you can use to just look up all kinds of cool DNA sequences for all kinds of organisms. So go to NCBI or OMIM and just search for a gene sequence. It's pretty cool. So wrapping up, we have accomplished our objectives. We define gene and genome. Gene, length of DNA that codes for some trait, influences a trait, genome, all the DNA, all the genes of an organism. We talked about genes versus alleles. Alleles are specific forms of genes. We talked about the cause and the consequences of sickle cell. It's a mutation. It's that one small, tiny little flip that leads to a difference. We go from glutamic acid to valine amino acid sequence in the protein, and that leads to that sickling of those red blood cells. We talked about how alleles are formed by mutation. We described that Human Genome Project, that international collaboration that allowed us to sequence the whole entire human genome fast and cheap. We talked about some of those ethical issues related to bioinformatics, and we talked about that crazy cool thing that is DNA sequencing. Good work today, my friends.